generates an individual like link for the live stream itself.
Cool. I see people popping on. Hello, folks. Where are you guys joining us from? Nice. And then I'm also going to pop over to our stream.
Tyler, just by the way, all the intros you made, that everyone took the meeting and everyone was awesome. So that was really super helpful.
Great, happy to hear that.
It's funny, like I feel like I'm kind of up. I'm starting to like learn the like gossip like in your domain.
Yeah, it's
because we are live.
It's been
recording.
The uptake of machine learning has been a little laggy in biology, but it's nice to see it start taking off, and now people are doing some pretty cool stuff with it.
It's super cool. Now I ask people, are you like small molecule or large molecule or biological, you know?
Yeah, and everyone's got different models and approaches. It's pretty cool.
Yeah.
Nice. Where's everyone else joining us from? I just see one person answering. All right, I see people popping into the stream and as well as in the Zoom. We'll give it 2 more minutes and then we're gonna go.
For that, I'm gonna put in the chat the direct link to the stream now. So if you want to share, feel free to send that link out.
Nice. All right, I'm gonna kick this off in about 1 minute. I see Han is joining us. Hi, Han. Han was one of our first speakers, and he was really good.
No math lesson from Charles this week?
No math lesson from Charles this week.
More
Hello,
coming
Charles,
soon.
you are here.
Yeah. Hey, Lavanya. Yeah, I just gave my final like defense of my thesis last week, so I can get back on the webinar train.
Nice.
Congrats.
Thanks.
All right, I'm gonna kick us off. Thank you everyone for coming. We really appreciate it. We know it's really late for some of you, and we appreciate that you still showed up here. This is our 5th time doing it, I think, and every time the quality of the speakers, the audience, and also just the whole environment gets better. We used to have technical difficulties, which we've slowly cut down on, so I'm very happy about that. And with that, I'm going to give this over to Lucas, who's going to introduce our speakers.
Nice, thanks, Lavanya. Yeah, I'm super excited about this. It's fun to see the wide diversity of speakers and talks. This week, for your entertainment, we have Tyler Shimko, who just defended his thesis for a PhD in genetics at Stanford, and then also founded a biotech startup, Trident Bioscience, which I'm super excited to learn more about. He's working on computational experimental technologies to expedite the discovery of useful proteins. And he's going to talk about one of my favorite topics, which is machine learning in biophysics. We also have Kaustav Sinha, who is a PhD and also an intern at Facebook AI Research. He is going to be talking about his paper evaluating logical generalization in graph neural networks, which I started reading just before prepping to do this, and I think it hurt my preparation for these intros. We also have Madison May, who I was just noticing his Twitter handle is Pragmatic ML, I believe, which makes me want to listen to his talk right there. He's a co-founder and ML team lead at Indico Data Solutions in Boston, and is going to be talking about long-term context in transformers. Definitely a hot topic. And then we also have Stacy, who is in some sense the top deep learning engineer at Weights and Biases. She's actually been with us since nearly the very beginning, and before that she worked on productionizing machine learning systems at a number of companies, maybe most excitingly on content discovery and recommendation at Flickr. And she's going to be talking about one of my favorite parts of Weights and Biases that really is her brainchild, which is benchmarks for collaborative machine learning and some of the applications, I hope. So super excited. I will hand it over to Tyler.
Thanks, Lucas. And I'll just share my screen and then we'll get started. All right. So thank you guys for the invitation to speak today. I'm really excited to tell you about some of the work that came out of some of my thesis, as Lucas said, from Stanford in genetics, with a heavy focus on biophysics and how we can apply machine learning methods and where they might fit in best to the overall experimental pipeline of biotechnology. So a little bit more about me. I did my undergraduate degree in biology at the University of Utah, focused pretty heavily on wet lab biology at the time, and then transitioned in 2015 to my PhD in genetics at Stanford, where I made the switch from a primarily wet lab background to a primarily dry lab or computational background, and started learning and applying machine learning methods to the problems that we were facing in our lab at Stanford. And then recently defended my thesis about 3 weeks ago now, and shortly thereafter founded a biotech company called Trident Bioscience to take some of the methods and techniques that I developed at Stanford and transition them to commercial applications for the discovery and refinement of useful proteins. And now I want to start my talk with kind of a very general overview, take you guys back to high school science class and the central dogma of molecular biology. And the central dogma states that the information flows from DNA to RNA to protein in this generally kind of one directional movement through transcription and translation of DNA into RNA, and then translation of the information contained in that RNA into protein. But that's really only half the story, this bit that you kind of get in high school science class. What happens to the protein is kind of lost in that process of explaining this central dogma. But that protein goes on to actually fold into a three-dimensional structure. And that three-dimensional structure is dynamic with time. It actually changes and the shape shifts so that it can perform different functions for the cell. In addition, you can get these post-translational modifications or chemical group additions to these protein structures that can further modify their function. And then this ensemble of all these different structures are what actually carry out the various functions for a single protein sequence with any given cell. And when I say protein sequence or DNA sequence throughout this talk, what I mean is that each of these molecules are composed of different monomeric blocks. So they're polymers strung together with different Lego-like building blocks in a linear chain. And it's the three-dimensional folding of that linear chain that really determines what the structure and ultimate function is going to be. And in the context of this talk, when I say function, what I really mean is binding. So the binding of proteins to each other, proteins to smaller peptides, so small chains of amino acids, or proteins to DNA or RNA. So you might be asking yourself, why are we really focused on this phenotype or this idea of binding from one protein to another protein or one protein to DNA? And the reason is that this is really how biology controls or tunes the kind of rheostat for different interactions and different cellular processes. And this biological rheostat can be defined by these two main axes. The first being affinity, or how tightly the different interactions take place, and the second being specificity, or how specific a protein recognizes its binding partner. And so if we divide this rheostat into different quadrants, we can start with the bottom left, where we have non-specific interactions, essentially proteins and DNA just bumping into each other randomly in the cell. If we then go to the other extreme, a really high affinity, high specificity interaction, we have something like antibody-antigen systems. These systems bind almost irreversibly to each other and with really high specificity. So an antibody can recognize its target protein in a sea of other proteins. If we then move down the affinity axis, we run into transcription factors, or proteins that bind DNA to regulate the transcription of downstream genes. So these are the proteins that play an integral role in determining which genes are expressed in which cells. And their binding is really important, but it tends to be pretty low affinity, but relatively high specificity. To fill out the quadrant, we can think about nucleosomes, or these proteins that the DNA in your cells is actually wrapped around. And these proteins play an integral role in actually packing the DNA into the nucleus of each individual cell. And they're a really high affinity interaction, but they have low specificity. They generally bind DNA with lower sequence specificity than the transcription factors. And as you might expect, these types of interactions are really important for a lot of different applications in biotechnology. For instance, one thing that you might be familiar with is CRISPR. CRISPR relies on the base pairing between the RNA as part of the CRISPR complex and the DNA sequence that it's trying to modify. And this has been commercialized by a bunch of different companies. Editas and Caribou are just two of these. Another type of interaction that's particularly important is antibodies. And with all of the COVID-related research, I'm sure you guys are probably more familiar with antibodies than you might like to be. But they're used in a variety of different applications. They're, of course, used as therapeutics, as you've been hearing about lately. But they also are the basis for a lot of different pregnancy tests. So these antibodies are recognizing components in the urine that are indicative of a positive pregnancy. And finally, we have transcription factors, which is where I'm going to start my talk, because these interactions are really important for a lot of applications in synthetic biology. And companies like Amyris or Zymergen, which you may not have even heard of, are really interested in how transcription factors are binding and regulating downstream genes and how they might be able to modify that to regulate gene expression for their desired traits. And so, as you might expect, given the importance of designing these different intermolecular interactions, or these kind of binding interactions, we might want a set of tools that would allow us to develop and modify different interactions to suit a particular need. So if we think about what tools we might need to actually design these intermolecular interactions, they kind of break down into 4 general categories. The first, and the ones that I think you guys came to this talk for, is a model of how these interactions are actually taking place. We then need a method to translate those predicted interactions, or the predicted mechanism of the interaction, into a way to actually test the hypothesis that's made by that model. And so what this looks like in a lot of wet lab biology techniques is generating a set of sequences, what we call a library, that you can then run through a certain experiment and test the actual outcome of the DNA sequence or protein being produced by that library of input sequences. Taking this a step further, we need to actually test that pool, or that library of sequences that we've generated, via some experimental method to get a measurement about how well they're performing a certain task. And then finally, and ideally, we would like some sort of method to update our initial model given the new experimental data. And hopefully we're able to build a closed loop system where we can get continuous improvement, a better and better model of how these interactions are taking place and the ways in which we might be able to change them to generate new functionally refined protein or DNA variants. And what I'm going to focus on for the purposes of this talk is kind of this upper corner here. I'm going to define what the model of these types of interactions might look like, and then start to think about how we might generate a method for making these hypotheses actually testable. And the reason that this is such a big problem in biology is that sequence space in general, and especially for proteins, is incredibly massive. It's really hard for us to fathom how large sequence space is and how quickly it grows. So for DNA sequences, there are 4 possible nucleotides at each position. And so as you get a longer and longer chain of these nucleotides, the number of potential sequences that that length of chain can give rise to grows as 4 to the L, where L is the length of chain. So it grows exponentially. For proteins, on the other hand, you have 20 choices per position. And so the number of possible sequences grows as 20 to the L. And so here what I'm showing is, given a certain protein sequence that we want to start with, we can mutate a certain number of positions to all 19 other choices. And here on the y-axis is the number of total mutants that we would be required to screen if we want to do this in a saturation mutagenesis or exhaustive fashion. And unfortunately, our most advanced experimental methods have a limitation of about 10 to the 11th sequences that they can screen in a single experiment. And so this limitation really defines our upper boundary for what we can do in an experimental wet lab technique. So to some extent, we must be selective in the design of these protein sequences that we want to screen in high throughput libraries. And to me, this really defines the best place for a home for deep learning methods, because we have these high throughput screening methods available to us. The cost of any given prediction being incorrect is relatively low. We're not using this to diagnose a disease or to immediately treat a disease, but the value of the informed search through pattern recognition is really high. So if we can recognize patterns in the existing data and use that to inform the sequences that we include in our library, it could be really valuable. And as a case study, I want to present the first project that I worked on during my PhD, which is characterizing and modeling the behavior of transcription factors and how they bind to DNA sequences. So as I mentioned earlier in the talk, these transcription factors bind to DNA, and they do so in a sequence-specific manner. And so when I say sequence-specific manner, I mean that the given protein is binding to DNA with a preference for certain short DNA motifs. An example of one is given here. So this is FO4. It's a transcription factor involved in helping yeast fungi respond to phosphate starvation, hence the name FO4. And the sequence preference is given here. And so the dominant nucleotides are these ones at the middle, CAC, GTG, also known, and you might hear me say EBOX motif. EBOX motif generally refers to this CAC GTG pattern here at the center. But unfortunately, or fortunately for biology, families of transcription factors often bind very similar sequences. And so here we have a related transcription factor, CBF1, that you can see also likes to bind this CAC GTG motif. And so this raises an interesting question, which is, if they have very similar binding preferences, do they actually just regulate the same genes? Are they fully redundant in their function? And if we look at the gene regulatory networks, so here, the transcription factor is given as these kind of central nodes, and all of the genes that they regulate are the ones connected to those central nodes. And what we see is that there's actually a very small amount of overlap between the genes regulated by FO4 and the genes regulated by CBF1. And so the question that we wanted to answer was, if these core binding motifs, so that CAC-GTG, the EBOX motif, is nearly identical, what differentiates the target binding sites for one transcription factor over the other in the actual genome of a living organism? And so what we have to understand is that these binding site sequences sit in different contexts within the genome. They're not just floating out as a 6 or 8 mer sequence in the cell. They're actually connected to a bunch of other DNA, and that DNA has its own sequence. And so our hypothesis was that it might be these sequences that sit outside of that main motif that actually govern whether the transcription factor is going to bind or not. And so we wanted to look at all possible arrangements, so all possible genomic configurations for these given EBOX motifs, and then measure the binding for FO4 and CBF1 against that target library. And the way we did this was we made this DNA library that we're able to use high throughput sequencing on. So we're able to actually measure and read out each of these individual sequences, where we fixed that EBOX motif at the center, CAC-GTG here, and then we allowed the bases next to it to be any of the 4 other nucleotides, so ACG or T. This generated about 1 million sequences in total and represents an attempt to characterize binding that's actually relatively large. So we don't have a lot of really good methods to measure 1 million sequences with the resolution that we wanted to. And so along with a postdoc and another grad student in the lab, I developed this BETseq method, which stands for binding energy topographies by sequencing. And it allows us to measure these transcription factor binding specificities in high throughput, importantly with energetic information behind it. So it has to be a biophysical measurement in order for us to be really interested in Polly's lab, my PI's lab. And so we wanted to do this with really high resolution and generate these physically meaningful values for each measured transcription factor DNA sequence interaction. And the way that we did this was we have a small microfluidic device that you can see here on the left-hand side. And this device is about the size of a postage stamp and has a bunch of these inputs and these hydraulically activated valves. So here's kind of a schematic of how the individual chambers within this microfluidic device look. And each one has what we call this button valve at the top that allows us to compress and actually stop the interaction of a transcription factor with a bunch of different DNA sequences. So what this allows us to do is bind transcription factor to the surface here, flow in DNA. We're going to select for the things that bind. They're going to bind. We're then going to trap them and separate them from the sequences that don't bind. And so this separation is what allows us to make a measurement, an energetic measurement of the affinity of different transcription factor DNA interactions. So then we can put each library. So here we have the bound and the unbound libraries go through high throughput DNA sequencing on a machine made by usually Illumina. And then this generates a library where we can actually order these sequences based on the preference of each given transcription factor to these kind of context sequences outside the eBox domain. And so here you can see we can potentially look at all of these DNA sequences in the library and rank them from highest affinity, which in delta delta G space. So delta delta G is just a measurement for how high affinity a sequence binds, and it has a physical meaning behind it. And the more negative that value is, the more tightly it's bound to the transcription factor. So more negative equals tighter binding, more positive equals looser binding. And so we can do this for the entire library of sequences. But unfortunately, as I said before, the methods that already exist for doing this don't really scale well to the million sequences that we have in our library. And the reason is because experimental noise really inhibits these energetic measurements. The two reasons behind that are that we can't calculate a binding energy for something we don't observe. So for 1 million sequences, you actually have to use quite a bit of sequencing in order to see every sequence in that library. And for low count values, so things where we only observe a few of those of that individual sequence, a few of those molecules, the values are extremely noisy, which make our ultimate energetic measurements really noisy. And so we thought about this for a while, and the solution that we came up with was to simulate this process. So the advantage of working in a biophysically meaningful space is that we know how these things should interact in a perfect world, and we can just simulate the experiment and see how well we do for different depths of sequencing. And the important thing to recognize here is that as you go to a deeper depth of sequencing, you're getting more reads, you're getting better data, but it's also more and more expensive. And so we simulated one of these BETseq experiments, and here on the bottom, we get the mean reads per sequence, so the number of times we're observing each individual DNA sequence. And then on the y-axis here, we're showing the median value for two different measurements of success. And the first is the fraction of the library that we're observing. We want to be able to see all million sequences. And then the second is the R² value with the true defined value that we put into the simulation. And we can see that as we start out on the left, we end up with these really kind of garbage data sets where we're only observing a fraction of our library, and the measurements that we're getting don't actually correlate that well with reality. On the other side, we have a really expensive experiment that yields beautiful data where we're observing nearly all of the library. And our data quality is really good as well. The R² value is really high with the expected values. But we figured that we might be able to sit kind of in this Goldilocks zone here in the middle where we're observing almost all of our library or all of our library, but the R² values might not be perfect. And so that really set up the expectation that if we sequence one of these libraries, if we perform the experiment at really low read depth, what we're going to get out is kind of not reproducible and is going to look kind of like a circular pattern. And in fact, when we ran the experiment, we ran it twice, 2 different replicates, and we do see this indicative circle pattern where each measurement does not correlate with the same measurement made in a different replicate. But since we had the simulation data in hand, we were able to say that we expected that, made the change to a higher read depth, and we end up getting better data out. But again, the problem with this is that if we run all of these experiments at really high read depth, it's going to cost us a lot of money. And we'd really like to avoid that. So we thought to ourselves, we might be able to use some type of modeling to extract and separate the signal in the data from the low read depth experiments from the noise. And so we wanted to see if we could do that. And the thought behind this was that we already use transcription factor binding models. In fact, I presented one earlier in this talk. So this motif that I showed you at the very beginning, it's important to ask how we actually got to that point. And it's through a very simple linear model that we can go from a bunch of different sequences to one of these motifs. So we ask, how do we get here? Someone previously had run a bunch of DNA sequences, did the same kind of enrichment step that we did earlier, pulled out a bunch of sequences that bound to the transcription factor, performed alignment, and then noticed that in all of the sequences that bound, there was a common pattern, this CAC-GTG pattern, and potentially this flanking nucleotide here upstream and downstream. And from that, they were able to generate what we call a consensus binding site. So a single sequence that is generally enriched in the binding sites for these transcription factors. But an important assumption in this methodology is that the contribution of each nucleotide to binding is totally independent. And so we didn't think that that assumption was that good. We wanted to ask, is this assumption of independence actually valid? And we knew, because the time that we were doing this work, neural network models were coming into vogue, we knew that nonlinear models would enable us to do the same type of modeling, but in an assumption-free fashion. So we wouldn't be forced into assuming that the contribution of each nucleotide in the binding site was contributing independently. And so we built ourselves a nice little neural network model. We feed in the DNA sequence here as a one-hot encoded vector, have a couple intermediate layers with nonlinearities, and then we're going to output the predicted delta delta G value and train on our experimentally measured values. And when you work on deep learning in biology, you run into a bunch of people that aren't too keen to trust that your magic neural network box model is actually accurate. And so you have to do something to prove to them that the data that you get out actually reflects reality. And we had another grad student in the lab, Arjun, who was kind enough to measure a bunch of the sequences that we had looked at on an orthogonal platform. So based on the same technology, but a much higher resolution technique that's much lower throughput. And what he was able to find and show was that if we take our unprocessed or raw data, the measurements that we had made without any type of correction, they don't correlate super well with the measurements that he had made in this orthogonal method. But if we first run it through our neural network and take the prediction output from that and compare it to his orthogonal measurement, they do correlate really well. So this gave us a lot of confidence that what we were seeing was good separation of signal from noise. But we also understood that in exchange for the flexibility of neural network models, we pay a price in interpretability. So we're not able to have that nice little motif anymore where we can say, okay, these are the contributions to binding. And so we were looking for a way around that. And we wanted to take a look at how well each of the existing models that were based on principled biophysical approaches can explain what's learned by the neural network models. So we basically wanted to ask, how far can a mononucleotide model, or that kind of binding motif model that I showed you in the very beginning, how far can that get us in explaining what the neural network has learned? And so if we plot the mononucleotides, so the linear model prediction down here on the x-axis and the neural network prediction on the y-axis, we see that for FO4, that linear approximation is actually pretty good. It explains binding behavior pretty well. But for CBF1, on the other hand, it doesn't do so well. There's a lot of variability for any given mononucleotide predicted delta delta G that can't be explained only by those mononucleotide effects. And so there's another method for doing this that adds a little bit more biophysical information, injects a bit more domain knowledge into this modeling process. And that's considering dinucleotide effects. And dinucleotide effects are just combinations of different mononucleotides at different positions. And these dinucleotide effects are really important for a bunch of different functions. But there's reason to believe that for FO4 and CBF1, especially, they might be important. And so what I'm showing here is the actual structure of the FO4 protein, which are these kind of blue squiggly lines up here, and then the DNA that it's bound to, the green helix down here. And I'm showing kind of two views of that offset by 90 degrees. And there's two different types of dinucleotide effects that we wanted to consider. The first were adjacent or nearest neighbor dinucleotide effects. These effects are a single base and the base pair right next to it. And these are really important for determining the actual three-dimensional shape of DNA. And so I know that I've been presenting DNA here as a linear sequence of these letters, but it actually exists in your cells as this three-dimensional helix that can bend, rotate, compact, expand. And a lot of those shape properties are determined by the nucleotides and how they stack right next to each other. On the other hand, we realize that these gapped nucleotides, or nucleotides that are present, dinucleotide effects that are not present in adjacent positions, might be really important because FO4 and CBF1 are actually dimer proteins. And so there's two individual protein chains that bind together and co-bind that DNA complex. And because these tails of FO4 and CBF1 sit within these grooves of the DNA, the contacts that are distal to one another might be actually important in the binding process. So if we add these dinucleotide features to our linear models and then compare them back to the neural network models, what we see is that for FO4, it nearly perfectly explains binding at this point. There's very little variation left. And for CBF1, it does a pretty significant job, indicating a significant improvement on the explanation of binding. If we add in all of the dinucleotide effects at this point, we can explain nearly all of the variability in the predictions from the neural network for FO4, and the same is almost true for CBF1 as well. So what we found from this work was that FO4 and CBF1 clearly bind in these two very different modes. So one has a strong preference for the mononucleotide effects, so it seems to be that each individual nucleotide is actually contributing independently to binding. But CBF1, that doesn't seem to be the case, indicating that for CBF1, there might be a much larger effect of something like DNA shape to how it binds. But if we want to ask the question why, the work that I've presented so far doesn't really get us there. And we're a bit limited going from DNA sequence to functional predictions directly via a neural network model. And the reason for that, if we go back to the initial presentation of the central dogma of molecular biology, is that instead of predicting each step along this process from protein to the folding and conformational dynamics, the post-translational modifications, and then binding, we're basically black boxing this entire intermediate step and going directly from the protein and DNA sequences directly to their functions. And it's important to note here that we're not explicitly modeling the structure of the protein or the DNA here. We're just taking a proxy measurement of their sequences for their structures. But why would we want to model the structure at all? Aside from the fact that I just told you that it was critically important. And I chose an example from the literature that I think illustrates this really well. And the principle here is that structural changes in the protein actually drive really important functional effects for the protein being targeted. And so here what I'm showing on the left-hand side is green fluorescent protein, also known as GFP. And this is a really important protein in molecular biology because if you hit it with the right wavelength of light, it glows green and it allows us to visualize proteins and motion within the cell. And on the right-hand side are two different nanobodies. So these are really cool single domain antibodies isolated from camelid species. So llamas, alpacas, camels, they have these really cool single domain antibodies. And a lab in Europe isolated two of these different nanobodies that bound this GFP protein. And they were doing some basic characterization of this, and they realized that as they added more and more of each nanobody, they actually saw a very real functional effect. And so the one on top in red, as they added more nanobody, it suppressed the fluorescence of GFP. But the one on the bottom, as they added more and more of it, it enhanced the fluorescence of GFP. And so they named these the GFP minimizer and enhancer nanobodies respectively. And all they really differ in is the site on GFP that they're binding. And what's interesting about this is that if we look at the GFP minimizer and enhancer nanobodies and start to ask, like, okay, what are the functional effects? What are the structural reasons behind these functional effects? We can look at the contact map of GFP. So if we look at all of the amino acids by all of the amino acids and then just highlight the ones that are in contact with each other, we see that when the GFP minimizer nanobody is bound, this is what the contact map looks like. When the enhancer nanobody is bound, this is what the contact map looks like. And they look generally very similar, which is to be expected. GFP's structure is pretty stable. But if we start to look at the difference between these two, we can start to see really important functional effects pop out. And so here, what I'm showing are in red, the enhancer specific contacts. So the contacts that are only present in the GFP bound to the minimizer nanobody or the enhancer nanobody, and then the minimizer specific ones bound to the minimizer. And so it would be really useful if we had some sort of model to actually predict the structural consequences of these types of binding interactions. So how might we go about doing that? If we think about two different amino acid residues, so here I'm showing two of the chemical structures, each for alanine, what we want is some model that takes as input those two residues and gives us back the probability of them being in contact with one another. And if we run this over all of the potential pairs of amino acids in a protein structure, we can generate this probabilistic contact map. And there are a bunch of different ways to do this. And in fact, AlphaFold has been very successful at generating these predicted contact maps for single chain structures. But one of the most promising, I think, is reframing this problem as a graph neural network problem. And so we can treat these proteins as a molecular graph because, as I said before, they're just a bunch of these amino acids chained together linearly. And so this, as I said before, is the structure of alanine. And what you might notice is that it looks a bit like a graph structure. You have your nodes and you have your edges that connect atoms with covalent bonds. And so if we apply graph neural network models, we might be able to actually model this process incorporating all of the atoms, not just the amino acids or the nucleotides like we did before for the FO4 and CBF1 modeling. And an important side note here is that using these atom level representations and not relying on the representations just of the amino acids as kind of elements of your alphabet is that it allows us a straightforward method for encoding post-translational modifications, or these chemical modifications that aren't amino acids, but are added to the protein after it's been translated. And so in general, graph neural networks operate over these graph structures through message passing. And I tried out two different ones in very early stage development, these graph attention networks and position aware graph neural networks to perform this exact task, where we're going to input atom level features and the covalent adjacency matrix, perform graph convolutions for the two different residues, and then output the probability of contact, generating that contact map. And so we don't just generate a single contact map, but actually we generate different types of potential interactions. And so those of you familiar with chemistry might understand why we might want to predict hydrogen bonds or van der Waals interactions. We actually predict a set of 8 different types of interactions. And so this work is still very early stage, but I want to present some really preliminary results, just overfitting one of these graph neural network models to a single structure to see if it has the capacity to do that overfitting. And so if we look at one small protein structure here, we can use the different types of interactions. So here, hydrophobic, hydrogen bond, van der Waals, and water bridges as our target. And then if we try a bunch of different models, we can see varying degrees of success overfitting to that single structure, with one of the models actually perfectly fitting the data, indicating that this might be a feasible approach for modeling protein structure, and importantly, modeling the interaction of different protein structures, so complexes of different proteins. And so what I've shown you here is that these graph neural network models are really powerful and directly applicable and directly translatable to chemical prediction tasks. That by using all atom models of protein structure, we're able to predict the effect of post-translational modifications, or things that are added to the protein after it's been translated. And that by using a deep learning model to do this type of prediction, we'll be able to do it in really high throughput, allowing us to generate those protein libraries that I mentioned at the beginning of the talk that would allow us to do really high throughput screening for functional proteins. And so if we combine these types of methods with existing methods based on physics simulations and experimental processes, we can kind of find the needle in the haystack a lot quicker than previously possible. And with that, I want to acknowledge a bunch of people that were really integral in this work. So Dan Lai and Arjun were the 2 members of the Fordyce lab that really helped me develop the BETseq assay. I got a lot of help from Anshul Kandaji's lab, in particular, these individuals listed here, on developing some of the early neural network models for this. The Leskovic lab has been super helpful in shaping my thinking about how to apply graph neural networks to the protein structure prediction problem. And then these two collaborators down here at the bottom, Yaron and Thomas, were very helpful on some of the other graph neural network and some work that I didn't talk about. And with that, I'll open it up for any questions.
Great, thanks, Tyler. So I see one question, and people both in on YouTube and people in the Zoom, can you please start asking your questions either in the Q&A or in the chat, and I'll come get you. The YouTube stream does run about 2 minutes behind, so we'll just take that into account. I do see one question here though, which is, what does the data for the neural network model look like?
I'll assume, but correct me if it's wrong, that you're asking about what the inputs and outputs to that network look like. And so the input to the network is just the one hot encoded DNA sequence. And so it's just a 4x L matrix where the individual rows represent A, T, G, and C. And then the length of that matrix represents the length of the DNA sequence. So that's the input. And then the output is the predicted delta delta G value. So that energetic value that we're actually measuring, we have a noisy estimate of that from our experiment. That's what we're using as the target for the training data.
Cool. This is not a question, but Daniel Cooper on YouTube says his mind is blown and he's really excited about the talk. Someone dabbed, so that was cool. I don't see. Oh, I see another question. Have you been using any of the state-of-the-art NLP libraries, Hugging Face maybe, to parse and analyze the DNA sequences?
So we haven't been using that. And what a lot of the other groups at Stanford have found really successful are actually the 2D CNN models. So treating that 4x L matrix, not as a sequence and applying NLP stuff, but applying a lot more of the computer vision techniques as a 4x L image, basically. So they're using that to train and test networks over full genome sequences. So the sequences that I was looking at here were only 10 base pairs. They routinely train on thousands or hundreds of thousands long sequences.
Nice. Cool.
Tyler, yeah. So really cool talk. I was wondering, so it sounds like you think that the sort of 2D CNN approach is going to work better for these one hot encodings of DNA sequences. Do you have any thoughts about why something like an embedding based model isn't what works there, even though that's been so successful in NLP? And yeah, also, why do you think graph networks are so successful for the other tasks that you've used them for?
Yeah, so I think that the convolutional networks versus recurrent networks or transformers, I think that a lot of that comes back to not necessarily that they wouldn't be successful, but that the 2D CNNs they generate predictions in a way that's much more interpretable for biologists and for bioinformaticians. And so we're used to thinking about these things in terms of motifs. And if you think about each kernel in a CNN as basically a motif finder, it's going to apply a weight to each nucleotide at each position. Then people have already done a lot of the heavy lifting of kind of generating interpretation methods for 2D CNNs. So I think a lot of it stems from that. You might see greater success using some of the NLP techniques, but the other factor is that the data tend to be really noisy. And the trick is that depending on who collects the data, which group, how they do it, you run into a lot of these batch effects and things that aren't necessarily normalized over different experimental techniques or different experimental protocols.
Cool. And then Han asked, what type of linear models or techniques were used in the sequencing, and how has modeling the sequences using linear models helped in designing your neural network?
Yeah, so the linear models basically use the same inputs and outputs. And so it's just essentially a 40-dimensional vector where we're flattening that matrix. So it's the same one hot encoded matrix and the same delta delta G values, same inputs and outputs as the neural network models. And I'll have to admit that it didn't really shape our thinking about exactly how to design the neural network model, we kind of worked from the neural network model backwards. And so we built a neural network model that fit our data and was validated via these orthogonal methods. And then we used the linear models to really interpret what the neural network was learning and how that fit with our assumptions based on the data sets that had already been collected.
Nice. And then I think the last question is, have you tried experimenting with graph neural networks in addition to convolutional neural networks? And if so, is any of this stuff publicly available?
Yeah, so the graph neural network stuff that I presented at the very end is still really early stages. We're not seeing the same level of prediction accuracy yet as something like AlphaGo or these recurrent geometric networks. So if you're looking for the literature, I would point you to either the AlphaGo, or not the AlphaGo, the AlphaFold paper, or a lot of the work from Mohammed Al-Qarashi's group at Harvard does a really nice job with some of the recurrent networks. They seem to be more accurate in general so far, but I think there's promise for the graph neural network techniques.
Nice. All right, I think that's it. Thank you so much, Tyler. This is really cool.
Of course, thank you guys.
All right, up next, we have Kostiv. So Kostiv is a research PhD intern at Facebook AI Research, and his interests lie at the intersection of generative language modeling and neural reasoning, which I'm super fascinated to hear about. Today, he's going to talk about one of the papers that he's written. His paper is called Evaluating Logical Generalizations in Graph Neural Networks. So, Kostiv, the floor is yours.
Thanks for the nice introduction. Just gonna share my screen.
Also, Kostiv is going to do an AMA at 9 a.m. in our Slack community this Friday. So if you have any more questions after reading his paper, please let us know and join the AMA.
Cool. Can you guys see my screen? Okay.
Yes, we can.
Awesome. So yeah, thanks for inviting me. And Tyler also, like, it was really great talk. And yeah, so let me go forward with this talk. So this talk is not essentially about only our recent paper. This is essentially about clubbing two similar works that I had. So I'm just gonna like go through that in this talk. So first, like a little bit of intro about myself. I'm a PhD candidate at McGill University and Mila, co-supervised by Joel Pinu and William L. Hamilton. And also I'm a research intern at FAIR Montreal. My research interests are kind of in intersection right now with systematic generalization, which is kind of a subpart of neural reasoning, and in discrete domains, such as in language or in graphs. And also I dabble a little bit into dialogue systems. And originally I'm from Kolkata, India, and right now I'm at Montreal. So, okay, so why care about generalization? So ML models are getting really good at feature extraction. So those of you have like seen Joshua's stellar talk in last NeurIPS. He talked about this topic of system 1 versus system 2 deep learning. So what is system 1? System 1 is where we are right now, where we have very good models which does feature extraction. So given a standard data set, that model like really performs quite well on that particular data set. And in the bottom right corner, you see that I have posted like a top 1 accuracy. This is taken from papers from Code. And this is a top 1 accuracy on ImageNet. And right now, the state of the art is just a little bit shy of 90%. So system 1 deep learning works really, really well. We have pretty good feature extractors. And also the number of parameters used by our models are like increasing like really, really fast. But what this talk is involving is I'm kind of trying to motivate towards system 2 deep learning. So this is reasoning, which involves inductive reasoning. Basically, when you learn some skills from your data set, you want to like apply those skills to some new data set or those data sets which are kind of produced from that particular data set. And you want to transfer your known skills, and you also want to remember your known skills. So basically, this is a slide I've just like kind of screen grabbed from Joshua's talk, where he talks more about system 1 and system 2 deep learning. So essentially, the system 2 or the future is to go towards a set of models which are kind of more logically driven. So essentially, whenever you're making like a prediction, you are making that prediction in a logically valid way, so that you can reapply your learned skills on top of a data set which is kind of derived from the original data set. So that kind of helps us to go towards a realm of out of domain distribution. Out of domain is kind of ill-defined. Like you can just say that, okay, I train on a subset of images involving cats and I want to test on images involving dogs. But in case of the out of domain distribution, it's better to present it in this way, that if your model can learn the integral skills or the integral components that is driving a particular task, can your models use those components and reapply them to a new task? So that example that I gave, it will turn up to be, let's say you train on a subset of cats and whether you can recognize a new breed of cat. So how do you know your model is reasoning? So there are a lot of performance metrics that we look at, specifically accuracy, or in binary classification, AUC on held out test sets. But that is it. Like that is the only measure that we are interested in, mostly in deep learning research. So right now there's a lot of push of building more interpretable models. We also want to build a lot more integrated test sets. So essentially you just not say that, okay, on aggregate, my model is performing 90%, but what does it show? Like, does it show that where your models are failing and how they are failing? And even if they are failing, like their failures should be kind of systematic. And also, how do you guarantee that your model is just not basically rote learning features, like given a distribution which is somewhat similar but not exactly the same, whether your models can learn on this kind of setups? And also, how do you evaluate the robustness of your model? So recently in natural language processing literature, a lot of work is coming up on adversarial attacks. And people have been shown over and over that with certain types of adversarial attacks, you can fool a lot of language classifiers and also a lot of image classifiers. And there's a lot of defense mechanisms are coming up. But again, like, how do you evaluate your model on that kind of robustness without having access to such kind of data? So the key properties in system 2 deep learning that I'm trying to propose is that we want to go towards systematic generalization. So essentially, we want to learn to extract the core skills of a task, and we want to learn to apply those skills to solve any task which can be produced from such rules. For example, if you are learning how to add, you're probably learning how to add like, let's say, single or double digit numbers. But let's say on test time, if I give you like n-digit number, then you should also be able to add, because it's the same skill. You're just applying it over a new distribution. So that is just a very trivial example, but in a lot of other cases, you want to be kind of able to extract the rules that are imbibed in your data set, and you want to make sure that your model like reapplies those rules. And you want to be invariant to noise, and to do that, your model must be logically valid. That is, if your model is like correct at certain examples, then it should be correct for those examples throughout a set of distributions. So let's say we take a simple example. Let's say an example can be a simple kinship relation game. Now, if I'm given you a text which says about a certain kinship relation between characters, you can, like if I ask you like, okay, how is Carol related to Justin in this case? Then you can easily say that, okay, Carol is the grandmother of Justin. Now, this kinship relations, we know like these rules that we already have learned from growing up. But the cool thing, what we do without realizing, is that we reapply these rules. So if I give you a long set of relations, and if I give you like a pen and paper, you can still solve that. You can still like reapply your own thinking to solve like, okay, this character is related to this character in this way. So basically, you can think of it as a graph also, like given certain entities and certain edges. So the edges are essentially denoting relations. And if you want to do relation prediction, that is, what is the type of a particular edge, then in this kind of a kinship relation game, you can easily do that by taking like a walk over the path and basically resolving that path. So one of my previous work is named Clutter, which is exactly this. So Clutter is essentially a diagnostic benchmark, which is created for evaluating inductive reasoning from text. It is exactly a kinship relation game where the model is essentially given a blurb of text, which contains a story. And it's a Q&A setup where you're given a question, and then your model has to give out an answer on all possible family relations. Now, each puzzle is built using first order logic. Now, that helps us a lot in basically making sure that all the puzzles are verifiable and the logic is built on a set of rules. Now, the generator is basically fed a set of rules that we know about family relations. And then the generator essentially reapplies these rules to create this intricate puzzle. So this puzzle has essentially two modalities. So it has the primary modality is the text, which we want the models to perform. And then there is a secondary modality is graph. That is, each puzzle also has an underlying graph. Now, this is also a kind of a diagnostic suite. You can build your own complex setups on top of this, and then you can see how your models are systematically generalizing. Now, just as an example, so this is an input document. This gets fed to the model, and then we have a query. So in this case, we have a query which is a pair of entities that we want the relation in. Interestingly, we didn't use a natural language question that you see in a lot of Q&A datasets, because there was a really nice paper in AMNLP 2018, which shows that a lot of these models, they do not even understand the entire context of the question. So let's say if there is a question is that, when did this event occur? The model kind of performs even well if you like cut off all the different words and still focus on the word when. This is like a really great paper by Zachary Lipton. I would suggest you all to read it. And so basically, like, we replace the question with the set of like queries. And then the task is simply to predict what is the relationship between them. Now, this seems very simple. And I have like heard from a lot of people that they are saying that, okay, this is very simple because you just have a set of rules that the model have to learn. And once the model learns the rule, and then it should perform well, right? So it turns out no. Why? Let us go towards a bit of the tasks first. So one of the tasks, or the primary task that we want to look at, is called logical generalization. So in logical generalization, you're given a set of like puzzles, which are kind of restricted by their length. So essentially, you're given a lot of examples, but you're restricting the length of this puzzle to only contain relationship combinations of length 3. And then you're testing on puzzles which has relationship combinations of length more than 3. So this is equivalent to the example I gave before, that if you know how relationships work, if I give you a long passage, you can still solve that given enough time and compute. So this is exactly the notion of system 2 deep learning, because the models have to think. The model have to like reapply their own skills over and over in order to solve this particular task. And also, we looked at a case of linguistic generalization. So essentially, we thought that, okay, a lot of NLP models are pretty good these days because they kind of learn a lot of features about the individual words. And then whenever they find the similar words, they kind of overfit on top of that. In order to make sure that the models are not doing it, for each puzzle, we have two representations of each puzzle. One representation is drawn, like basically each puzzle is a graph, and then you're applying a layer of language on top of it. And this layer of language is what we are collecting through Amazon Mechanical Turk. And essentially, we are splitting the placeholders of the Amazon Mechanical Turk into training and testing. So this becomes really challenging for the model because the model has never seen the exact syntax before. But if the model understands how relationships work, then it should be able to generalize better in this kind of setup. So, and also a third cool thing that we had in Clutter was robustness test. So as I said that since these are built on logic, you can easily verify whether your logic is sound or not. So let's say given in this example, if I have given you a family relation tree like this, and let's say I want you to answer what is the relationship between A and D. So then there can be two possible paths in the supporting fact. And then your model has to understand, okay, since there are a supporting fact, I have additional information to do well. While if I have a tree which has a dangling path, that is kind of a confounder. And that confounder is like used to make sure that, okay, your model does not perform well, but your model has to understand, okay, I do not want to go through this path. And then there is a disconnected fact where you have a edge which is entirely disconnected from your like your current resolution path, but then your model has to also disregard that. So these are really simple but very powerful robustness test that we have in Clutter. And so let's go towards like what are the models we initially used. So we essentially thought, okay, we have made the task really difficult for the text or the natural language understanding models. We also need to provide a benchmark for a model which do not have to do the parsing. So we used a graph attention network, which worked solely on top of the raw graphs. And for models that have access to the text, we used a vast number of models, including transformer style models such as BERT and BERT LSTM, which is essentially a layer of LSTM on top of BERT, on top of frozen BERT, essentially. So how does the models fare on generalizability? In the plot on your right, you see that the generalizability or the accuracy during testing drastically falls down when we show tasks of more and more length. While if we provide more training data, like if we provide the model training data of involving 4 number of relations, then the model like performs somewhat better. But the graph neural network performs the best when given a lot of additional data. But still the generalization towards like extreme generalization, what I say, like towards the end of the number of 10 relations, then all the models kind of falter. But graph neural networks are really robust. When we use the different robustness test, we found that the graph neural networks almost always outperform all the text question answering models, which is understandably so because the graph neural network do not have to do the semantic parsing. So semantic parsing is one problem, but still the graph neural networks falter on the length generalization problem. So basically, Clutter was based only on 15 rules. And these 15 rules were kind of a very boiled down set of family relation rules, which we used in order to make sure that the puzzles do not have any ambiguous relations. Now, how do we evaluate the adaptation of the models to different rules? Now, why is adaptation important? It is important, let's say, for example, in recommender systems, you have your model which is trained on production for certain products, and now your company wants to sell some other product. Now, you have to adapt on this new data of products because you have to learn the relationship between these new products. But if your model already knows how to pick up these relationships, it will be able to pick it up really well on this new data. And also that would be really useful later on for smart dialogue agents, which will be able to like converse with you based on some prior information that you have already shared with it. Also, we want to know in system 2 deep learning that how do we remember old rules? For example, when we are going forward to newer and newer tasks or newer and newer rules, do we still remember the old rules that we learned before? So these are the 3 key requirements for logical generalization. We want to inductively reason to unseen compositions, which is compositionality. We want to learn to adapt new rule set, which is multitask or even meta learning. So these are 2 different sets of problems. In this paper, we only looked at multitask, but meta learning can easily be done on top of this data. And we want to see how does the models remember the learned rules? So given a new set of rules, the learner should not forget what it has learned before. So now we come to the current paper, which is again another data set, but this data set is focused purely on graph, because in the previous paper we found that the graphs have performed so well. So then we thought, okay, let's see how far we can stress test graph neural networks. And this is a multi-purpose, multi-relational graph dataset, which is built using the rules of first-order logic. But unlike Clutter, so Clutter was only built on top of 15 rules, and still it was challenging enough. In this work, we went a bit more crazy. We generated a large bunch of rules, and then we said, okay, once we have generated this large bunch of rules, we want to basically sample rules from this large set in order to create different worlds. Now, different worlds essentially mean different buckets. So each bucket contains a set of rules, but these set of rules are overlapping with each other. So let's say there could be a scenario where there are two worlds. The two worlds have the exact same set of rules except one. So then that two worlds are kind of 95% similar based on the number of rules that are available. So that gives us a really nice way to define similarity. And similarity is important because it helps us to define how far your task is going out of the current distribution. So when people are interested in out-of-domain distribution, the standard problem that people face is that there is no such metric on different data sets to define what is exactly an out-of-domain distribution. But if you define your data set based on standard first-order logic, you get a really nice interface to do that. And that's exactly what we did in GraphLog. So essentially, each world, so these are like W1, W2, Wn, these are different worlds. And each world consists of 5,000 graphs in training, 1,000 in testing. And then these different graphs, so essentially we had a total of 57 different worlds. And the number of graphs are also quite large because there's 5,000 in each of these worlds. And also each world has its own world-specific graph. So basically, what is a world-specific graph? So essentially, when we are generating graphs for each world, we first generate a huge graph. So this graph is basically generated by applying all the rules over and over and over and over. And when you have this huge graph, you are sampling smaller and smaller graphs from this huge set. And so these smaller graphs essentially become your training set. But then another sample of this huge graph is your context representation. Now, why is context representation useful? Because if you want to do multitask learning, if you do not have any sense of context, then the models would essentially not perform well at all. Because the model is essentially learning some objective function in some task, and then it is switching to another task, and then it would drastically not perform at all. So what would be a context here? So since we are looking at a relation prediction, the context are essentially the relation embeddings. And since we are providing a world graph, a model can take that world graph and then output certain relation embeddings that would be useful for that particular world. And then the other models can take that particular embedding and then work on top of it for that current world. So it's very easy to compute similarity. It's very easy to compute out-of-domain distribution. And so with this help of 5,000 graphs in each world, number one, you can perform supervised learning as usual. So the supervised learning essentially consists of the compositionality test, because in the test set, you are essentially figuring out the unique compositions of relations which you have never seen before. Like you are given to predict the edge or the relationship between a source and a sink, whereas the composition or the walk between the source and a sink is unique. And then you can do multitask learning. On top of all of these worlds, you can train your models to do multitask learning. Essentially, that means that you're training on one world at one epoch and another world, another epoch. So essentially, you're trying to learn all the world information at the same time. Now, in the beginning, we said that we had this set of rules generated in the beginning, and then we are essentially doing a subset on this set of rules. So that means by doing a multitask learning, you will never go into the region of invalid rules, because all the rules are generated at the same time. So all the rules are already made sure that all the rules are consistent with each other. So by doing this kind of multitask learning, you won't have to worry about whether the rules that you're learning in world A are inconsistent with the rules that you're learning in world B. Sure, these two rules can be very different, but if you're putting it together in the same model, there wouldn't be an inconsistency issue. And finally, you can do continual learning. So this is highly exciting, because our work is one of the first work in graph datasets which has this like added objective of continual learning. And there's a lot of recent work, exciting work coming up in continual learning, where people are trying to figure out how to basically see that whether your models are learning to remember on previous tasks or not. Given this set of world splits, we can define each task as individual world split. And that is our like proposal of doing continual learning purely on top of graphs. So before I jump into the results, so I want to like talk a little bit about the models that we use. So essentially, we used a lot of models involving RGCN, which is a popular model used for relation prediction, and graph attention networks, and GCNs also. Now, as I said, when you're trying to do multitask representation, you want to have a context. And how do you get this context? So basically, we divided the model into two parts. One is a representation function, which takes into input the world model, the world graph. So essentially, each world has a unique world graph. And this model ingests the world graph and outputs the set of relation embeddings, which are contextual for that current world graph. And then another function, which is the composition function, which is also another graph neural network, that takes these relation embeddings, and it takes all the different 5,000 graphs and tries to predict the missing relations on top of it. So in this kind of domain, we took a lot of combinations of available like composition and representation functions. So a very simple representation function can be just a parameter, or like just a weight lookup, or basically like an embedding lookup in terms of an NLP. So why that wouldn't work? Because an embedding lookup is essentially learning to like backpropagate to each relations from all different worlds. So it basically does not learn any world-specific information. So on top of that, we can optimize that by using a GCN and a graph attention network as our representation functions, which take into account the world graph, and then gives you a set of weights, or basically a set of relations to work on top of. So, and then the composition function is essentially a model which can predict relations. So this model has to operate on the edges, and RGCN was one of the really famous models which works on top of these relation prediction tasks. We had to modify GAT. So for the clutter task also, we modified GAT to attend over edge representations. So originally, GAT, or graph attention networks, is basically during the message passing update, the graph attention network attends to the different messages that are coming in. But in our modified edge GAT, the graph attention network is attending to both the messages that are coming in and also to the edge representation of the source and the sink. So this EdgeGAT is also the same model that we used in Clutter, and that's why we use it here also. So how do the models fare on generalizability? So when we do supervised learning, we find a nice like a separation of difficulty of different worlds. So this difficulty is essentially like a separation based on the number of unique composition or descriptors available in each world. So in some of the worlds, the number of compositions are not enough. So essentially, for each of the graphs that we generate, these graphs also has the set of noise that you have seen before. So in Clutter, the types of noise that we generate, we also use the same types of noise here. So that adds a lot of other difficulty. And these procedurally generated worlds, when we do a supervised learning, then we have a nice like proxy, which is the supervised learning scores. And these scores are basically very similar to each of the different models. And that gives you how much easy or difficult each world are. Now we do multitask learning. Now in multitask learning, we increase the number of worlds, and then we see that we hit a saturation point at 20 worlds. So that can be an outcome of the number of parameters that we had in our model. Like if we increase the number of parameters, we can increase this number of saturation. But if you see the accuracy, even at 20 worlds, the highest accuracy is 60%. Whereas in individual worlds, the accuracy goes towards 80 to 85%. And then we also did multitask learning on the easy and on similar and dissimilar splits. So essentially, S is the similar split, where we took the worlds which have a similar set of rules, like which are not differing too much from each other. And D is a set of worlds which have very dissimilar set of rules. And then in both of the cases, we find that graph attention network and edge-GAT combination is performing the best out of all different baselines. So now we look into multitask adaptation. So in multitask adaptation, we train using the multitask objective, and then we adapt based on the weights learned from this multitask objective. And we want to see two things. First, we want to see the final performance after adaptation. And then we want to see the adaptation speed. And during this, so just a clarification, this is not traditional meta learning. This is just simple multitask learning, and then you're adapting on the weights that are learned on the multitask learning model, which is very similar to what we do in transformer models, where you do pre-training, and then you basically fine tune on your given task. So in the graph you see on the left, on the orange like bar is one that we represent, which has very dissimilar set of rules. And the blue bar is the one where we have a very similar set of rules. And what we see that for most of the models, when we do multitask training on dissimilar set of worlds, then the final adaptation is much better than when we train on similar set of worlds. So this is a very nice finding, and that kind of correlates with a lot of pre-training literature, that whenever you're doing pre-training, you should try to incorporate as much dissimilar set data points as possible in your pre-training pipeline. And similarly, we see that when we look at the gradient updates, the models which are trained on dissimilar set of worlds are faster in adaptation. And finally, we come into the results of continual learning. In continual learning, we have two plots. So the blue plot is essentially the current task performance, and the orange plot is the performance of the mean previous task performances. So the mean of the previous task performance is basically falls down rapidly, and also it's pretty bad. And that is where we need to improve a lot in graph literature. And this data set essentially gives you a very, very challenging benchmark to do continual learning. So in the second plot, I'm showing the continual learning results when we ordered the worlds in terms of its difficulty. So recall that we got a difficulty score while doing the supervised learning training. So we ordered the models according to the difficulty score. And then we see that the single task performance is according to the model, but then the mean of previous task performance is also pretty bad. And that needs to be done. So basically, there is a lot of grounds to cover in terms of continual learning. And we believe this is a really, really challenging benchmark for it. So essentially, the key takeaways in GraphLog is that training on diverse data sets improve the generalization performance, and pre-training on diverse data sets also help to adapt to newer data sets. And pre-training on difficult data sets improve the model adaptation ability. So difficult also includes dissimilar data sets. But if you read the paper, we also have a topic on how easy and difficult training works in these kinds of setups. And we essentially have a lot of work to do in order to perform true lifelong learning in graphs. So in the conclusion, I show you like basically a snapshot of the two data sets that I've been worked on the last couple of years. And essentially, one of them focuses on the modality of graphs, while the other focuses on the modality of text. And for both of them, I have like released blog post. And for GraphLog, we have released an API, which is a really easy to install pip installable file. We also are looking to release the similar kind of API for Clutter as well. And using this API, you don't have to worry about data loading. So I provide data loaders for you and easy to use experiments, and also easy to use scripts written in PyTorch Lightning for you to like run your experiments just like that. And we have a generator module for both of these data sets. The generator module for Clutter is already out there. We will be doing a lot of like improvements based on user feedback on top of it. And also the generator module for GraphLog is coming out very soon. So yeah, that's mostly it. Thanks to my awesome collaborators, Shagun Sodhani, Jin for Clutter, Joel and Will, my supervisors. And also thank to my amazing colleagues at Mila and Fair Montreal for all of their support. And if you have any questions, you can ask me now, or you can like send me a mail. Yeah. And also you can like join our logical ML Slack, which I've opened to answer more questions. Thanks.
Nice. Thank you so much. So we have 2 questions already.
Yep.
So I'll go ahead and do that. And then the YouTube folks, you can start asking your questions on YouTube and we'll get those for you as well. All right. So Mohammed asks, what if the relation is not defined from the text? Would the model still be forced to predict the value of the edge?
So yeah, that's a very good question. We made sure that the relations are always defined in the text. So that kind of error do creep in if you are working a lot with Amazon Mechanical Turk. And it's a very messy situation out there because you have to do a lot of like cleaning on the data that you collect. So we made sure that the data that we collected or the templates that we collected has all the relation identifiers. And without them, you cannot like essentially solve the puzzle. So we made sure that you have the relation IDs.
Nice. And then the second question is, rules are super interesting. Is it possible to inject custom rules into experiments to see how they perform? Is it also possible? Oh, go ahead. Yeah, just answer that one.
Yeah, so that's a really nice question. And yes, it is possible. For both of the data sets, for Clutter, you can just easily like edit a YAML file and do the generations. So in the YAML file, the way I define the rules is it's essentially a first order horn clause. So the horn clause is essentially a rule which has like a body and a head, where the body composed of like 2 entities and a head is the output of that entity. So you can inject rules in that fashion. In case of GraphLog, we are essentially generating rules on the fly. So we are not using a single YAML file. So in case of GraphLog, what you can do, you can tweak with that rule generation parameters. So there's a lot of parameters which says that, okay, how many symmetric rules we will have, how many like invertible rules we will have, and so on. You can increase the set of rules like to a lot. Like if you want to do massive experiments, you can just play with them. Like both are essentially data set generators.
And then a related question, is it possible to extract rules created and understood by the model?
Yes. So your question is whether it's possible to extract the rules that are understood by the model. So this is a very interesting research direction on its own. For graph neural networks, there is like several papers which look exactly at this problem, like using graph attention networks. They try to like predict which of the edges that the attention are like kind of highest. And those are the edges that the model is kind of perceiving. We haven't done that kind of experiments in here, but that would be a really interesting way to look at.
Nice. So those are all the questions, but
I had a follow-up or related question actually to Mani's. So you presented for the Clutter work, you presented some stuff about generalization to relations, like longer sequences of relations, reasoning to longer sequences. So back in the early 90s, some folks did work with RNNs to do addition. And you used addition as an example of a similar problem. And they looked at how long, like how far out could they, like how long digits could they add together. And they were able to extract from the RNN an actual dynamical system that the RNN was implementing approximately that could do addition to basically infinite precision. Basically, you know, like it was they could find a set of weights that could then do even better than the network had done on the task. So that used some of the sort of specific ideas of RNNs, but I'm curious if for some of these like logical ML things that get closer to you know, traditional computation, if there's any hope that the networks that we're building and learning, that we might be able to extract back out from them an algorithm, something interpretable, something that's like compilable down to traditional code, or whether you think that that's a gap that can't be crossed.
No, that is also like an exciting research direction, what people do in program synthesis. So essentially you are trying to like generate the programs which you are essentially you're trying to optimize the programs that you generate and on your particular task. So there is a lot of research going on there. And I'm not super familiar with the research literature, but the way you mentioned it, it would be like possible. Like you essentially generate a program that will do kind of a recursive computation on top of this data set. And that recursive computation model is basically what we need. But that is also like super hard. And I would also like term that as system 2 deep learning. And that is what we want to move towards.
Nice. We have one more question from Heinz. For those who are not familiar with graph neural networks, do you have a sequence of papers that you recommend that people read or other resources?
Sure. For graph neural networks, I would suggest to start from the basic GCN architecture by Thomas Kiff. And then you can look at Peter Velikovic's graph attention network, which is really popular. If you want a condensed survey, so my supervisor, Will Hamilton, will be releasing a book very, very soon. So we are super excited. Like we actually like use that book for our course this semester. So that book will contain like a really, really nice in-depth overview of the entire theory and the applications. So just look out in his website. That's all I can say.
Is there a publishing deadline for that yet? Or like when is it coming out? Do we know?
That I don't know. I have to ask him.
Cool.
But it
Cool,
should be coming out soon.
Nice.
You can actually find the online draft on Will's webpage. So Will's webpage, Will has a link towards his course, which is Comp 767, and in that, or it was 766, yeah. So basically, in that course, you will find the list of notes, and those notes are ones that he will compile towards his book.
Nice. All right, those are all the questions I see. Thanks, Charles, for sharing the link. Costa will be in our Slack community at 9 a.m. on Friday, answering more questions. We'll dive deeper into his paper. It's going to be a great time. Yeah, thank you for coming.
Thank you so much. Thank you.
Of course. And up next, we have Madison. I'm super excited to hear from him. So Madison is a co-founder and the machine learning team lead at Indico Data Solutions. They're based out in Boston. Madison is going to talk about long-term context in transformers. And I'm super excited to have him on.
Hey folks, pleasure to be talking tonight. So like Lavanya said, we're gonna be talking about long-term context in transformers. And the goal of this talk is to give a pretty shallow look at a breadth of diverse ideas for long-term attention. I want to just make sure you're aware of some practical tips and tricks that you might be able to apply to your own problems that might even be outside of the NLP field. And in general, it's going to be an engineering lens into NLP with a focus on computational and memory cost. And this is just because of my own background. So this is the result of a couple of weekends spent diving into this particular subfield and doing a bit of writing and blogging on the topic. And if, like me, you're based on the East Coast, getting a bit tired, having some trouble staying awake, I've hidden tiny images of Bert on every slide. So you can use this talk that way instead if you prefer.
We appreciate that. Also, I'm so sorry. I didn't realize that it's like 10 p.m. for you. We should have had you go.
Oh, not at all a problem. So why might we care about long-term context? Well, first of all, if we're interested in text generation, we might want to remember characters or key plot elements in a story for more coherent text generation. We might want to keep a consistent style throughout longer generated texts. Or if you're like me and you're in industry, you might be interested in processing long documents that reference previous content. For instance, something like a legal definition. So most of you at this point are probably familiar with BERT. BERT uses this concept called self-attention, dot product self-attention, to route information between tokens. And just for kind of a brief overview, we take our hidden state from a layer, we project it out to a set of queries, a set of keys, a set of values. We compute a measure of agreement between queries and keys via a matrix multiply to get our attention matrix. Then we normalize these values and we use them to weight the values that were projected out from our hidden state. And the problem we're going to be focusing on today is this matrix multiply and this large attention matrix. Because as our sequences scale in length, it's these two steps that become particularly problematic in our network. Like I said, this is going to be kind of an engineering lens into the problem. So let's talk a little bit about runtime complexity. So for a simple dot product self-attention, we have as an input for a natural language processing problem, a tensor of shape ND, where N is our sequence length and D is our hidden dimension. And we want to compute the dot product between every possible token pair. So to do a little bit of simple math, we need N by D multiplies. So for typical choices of N, like in BERT defaults, we have 512 and 768. That amounts to about 200 million multiplies. But if we look at a context length of 8,096, so about 16x longer, or exactly 16x longer, we now have 50 billion multiplies required to compute our attention matrix. And more generally, we have an operation that is big O of n squared d. And it's kind of worth noting that 8,096 really isn't that long in practice. Like long books are hundreds of thousands of tokens. So we have quite a long way to go. Memory complexity also becomes problematic, and in some cases is more problematic than the computational cost of dealing with long sequences. So here we'll talk about multi-head attention, where we're looking at a couple different lenses into the same sequence. For multi-head attention, we have a tensor of shape k, the number of heads, by sequence length that represents the attention between the source sequence and the target sequence. We'll have some nice visuals a little bit later. But just for some quick back of the envelope math, we're going to have to store 12 by 512 by 512 floating point values, about 3 million floating point values for vanilla BERT, or about 12 megabytes of activations. When we scale up to longer context, this becomes 3 gigabytes, or 800 million floats per layer. So if we were to have 12 layers of these sample networks, we would need about 144 megabytes per example for BERT. And for the longer context setting, we need about 36 gigabytes per example. So already it's pretty clear why we have a problem. This big O k n squared value quickly explodes, and we can't feasibly store 36 gigabytes worth of activations in our model during training time to allow for backpropagation. So let's start diving into some of the details of how we might resolve this issue. So first up, we have the cheap engineering solution, which is instead of processing the full document at a time, let's break it up into smaller chunks. This is kind of good first go-to. The problem is, within each chunk, we have no way to use information from other chunks. And if you're looking or you're interested in filling in the blanks for tokens on the edges of a given chunk, you might not have the necessary context to perform that task well. So these edges are a problem. One possible engineering solution to this problem is simply to compute overlapping chunks and redo some computation. So this is twice as expensive because each chunk overlaps with half of the previous chunk. But it helps to partially resolve the issue we had previously, which is we have some tokens on the edge where we don't have a lot of relevant context. So in this setting, we're actually just using the predictions from the middle portion of the chunk as the outputs for our entire sequence. If you prefer kind of a more machine learning approach to solving this problem, I'm going to be talking over 4 different paper selections. We're going to kind of fly through things, but hopefully there's some relevant ideas that you can apply to your own tasks. So the sparse transformer, the compressive transformer, a model called the reformer, and the routing transformer. First up, the sparse transformer. This is by a group of folks out of OpenAI, Ruwan Child et al. This diagram is a visualization of which tokens a given query token attends to in an input sequence. So our query tokens here are denoted by the dark blue values, and our target tokens, which we're attending to, are denoted by the light blue values. So this is an example of causal attention, like you might use in an autoregressive language model. And this is kind of our baseline. So the colored values represent roughly the number of floating point operations to get done. And this is an alternate view into solving the same problem that the OpenAI team calls sparse attention. And there are many different kinds of sparsity, but in particular, they call theirs block sparse attention. And they've chosen to break up this dense attention operation into 2 factors. So the first factor we have is this block local attention, where each token only looks at a small local window around it itself. And secondly, we have long-term attention, which is looking at specific tokens throughout a much longer sequence. So we've introduced some dilation into our attention. And what you find is that the network can route information to these aggregator tokens with the light blue squares. And then in the next layer, the subsequent layer of this model, you can attend to these aggregator tokens to use information from that aggregator token's local block. So the cool property of the sparse attention model is that in 2 layers, you can hypothetically route any information between 2 tokens. So first by local aggregation, and then by attending to these aggregator tokens. And this actually has some empirical motivation. If you have ever read the paper, What Does BERT Look At? An analysis of BERT's attention, they dive into some of the patterns that BERT attention heads have learned. And one of the 2 trends that they find are attending to separator tokens and attending to periods. So BERT has naturally learned that there exist some aggregator tokens in a sequence where information is kind of summarized. And by attending to those summaries, you can route information over longer distances. So there's some empirical grounding here for this approach. They also had to overcome some computational challenges in order to make this approach practical. One of the papers they looked to is called Training Deep Networks with Sublinear Memory Cost by Chen et al. And the key idea of training deep networks with sublinear memory cost is that on the backwards pass, rather than storing every single one of our activations, we can locally recompute a subset of the activations using checkpoints. So specific activations that we chose to store. And we go from those checkpoints and we recompute a portion of the forward pass, the other intermediate activations, so that we can apply backpropagation. So it's really just a strategy to avoid caching all of our activations to compute our backwards pass. And we really need these large batches in order to efficiently use GPU and TPU hardware that relies on being able to efficiently parallelize things. And one thing you'll observe, if you look at the particular activations the sparse attention paper chose to checkpoint, they chose not to checkpoint this k by n attention matrix that was so problematic in kind of our initial back of the envelope calculations. So they chose instead to checkpoint some of these smaller activations of the network. If you want to do some quick math here, typical cost of a gradient update is 1 forward pass plus 1 backwards pass. When we use gradient checkpointing, we have to do 1 forward pass plus 1 backward pass plus another partial forward pass to fill in the missing gaps. So worst case, this is something like 50% slower for the same batch size, because worst case, we'd be computing 2 forward passes and 1 backwards pass. But oftentimes we can save more than double, or we can more than halve our memory cost. And by more than having our memory cost, we can more than double our batch size. And by increasing our batch sizes by more than a factor of 2, we can sometimes actually see speed ups, even though we're doing more computation per update. So the key ideas expressed in this paper are that we can factorize dense attention to 2 sparse operations that we run in series and still hypothetically allow for attention between any 2 tokens. We get O n times square root n memory and runtime complexity. So we've gained a factor of square root n. And if our n is something like 8096, this is pretty significant. This is something like a factor of 90. And we can checkpoint activations to further improve our memory use and scale out to even longer sequences. So memory use is very frequently the bottleneck when we're dealing with very long sequences. Next up is the compressive transformer. So this is a paper by Ray et al. These are folks out of DeepMind. The compressive transformer is largely an extension of Transformer XL with one key addition. We're going to talk about Transformer XL, and then we're going to talk about the novel contribution of the compressive transformer. But I think this GIF is kind of useful in illustrating the 3 main components of the compressive transformer. The first being the current chunk we're looking at, which is labeled sequence in this diagram. The second being memory, which is simply the previous chunk that we processed that's gradually moved back in a queue. And finally, we have compressed memory, where multiple memory time steps are compressed into a single state. So this is a lossy operation, but we want to learn to lose as little useful information as possible. So we'll talk a little bit about how we optimize for losing as little useful information as possible. First, let's talk about some of the contributions of Transformer XL, attentive language models beyond a fixed length context. So as we discussed previously, one of the easy solutions for dealing with long sequences is to chunk them up into smaller chunks. The problem with this is that you have no attention in between segments. Transformer XL proposes that we could cache these prior activations and attend to them, but not actually compute the gradients through this operation to save some time at training time. And this cheap trick gives us this neat property that when we have a model that is n layers deep, every layer, we can add some additional context to our model. So you'll see this token, instead of having a context length of size 4, has a context length of something like 10 here, because every prior layer can attend to tokens that might have occurred outside of its context in the prior layer. Secondly, the Transformer XL paper introduced this concept of relative positional encodings. One of the problems with representing long documents in chunks is that when we're using absolute position indices, so each position in a sequence is just labeled by its index, and we have an embedding for each index, chunking resets the counter at 0. And learning representations for the beginning of a chunk really isn't that meaningful for long sequences. Ideally, we'd want position index 0 to mean something like the start of a document, or honestly anything that doesn't rely on kind of taking an arbitrary modulo 512 or modulo chunk size. So resetting to 0 doesn't really make sense when we chunk up documents. Relative position embedding instead looks at the difference between two different token indices and learns a representation for each relative difference. It also does some more weight sharing, but for the purposes of this talk, this is the key contribution. And when we talk about relative positions, we have a more sensical interpretation for long sequences. The key contribution to this paper is actually this compression network. This compression network is a tiny little MLP that takes a few memory states and compresses them into a single compressed memory state. So it has two parts. The first is this compression module, which is this tiny little MLP. They tried some baselines like mean pooling, max pooling, the most commonly used memory states, for instance. But they found that the best objective was actually to have pretty much a secondary model that tries to reconstruct the attention matrix over these memory states from the single compressed memory state. So the objective here is we want to mimic the attention over what we've compressed. And this is more or less trained as a separate network, so the loss doesn't blend with that of the main network. And this loss, the secondary loss, allows them to achieve better results than some naive heuristics like mean or max pooling over this sequence. And we can theoretically attend to tokens compression rate times n layers times context size tokens away. So we get a factor of compression rate out of this operation over transformer XL. One often overlooked contribution is simply the fact that they also introduce a new data set for this task. Oftentimes people benchmark on Wikitext 103, LM1B, and Wiki 8. These are some of the data sets people use for longer term sequence modeling. But to a certain extent, we're kind of outgrowing those data sets. And so DeepMind team built up a new data set called PG19 based off of Project Gutenberg, which is an open repository of public domain books you can find at gutenberg.org. On average, these documents are 20x longer than the LM1B benchmark, and it's about twice as large. So dataset contributions are hugely important to the field, and I'm really looking forward to the research that comes out of new papers that use this better benchmark. So the key ideas here are this concept of using relative positional embeddings, a segment level recurrence mechanism, even if we don't backwards propagate, even though we don't back propagate the other chunks, an idea of compressive memory, and the application of a better data set for more rigorous research. Next up, we have The Reformer by Nikita Kitev et al. And I believe he's actually an undergrad, so this is extremely impressive work. At a high level, The Reformer takes your input sequence. It applies locality-sensitive hashing to bucket the input sequence up into these different colors. We sort by color. We attend within each color here. So we chunk things up and we attend within elements of the same color. Chunking is kind of just an implementation detail here. So for the most part, you can ignore that. First, they found that the idea of having a separate query and key isn't entirely necessary in order to attend over a sequence. So if you actually tie the weights of the key and the query, you get similar performance. And they use this as inspiration for their locality-sensitive hashing operation. If you're unfamiliar with locality-sensitive hashing, the idea is to, in this case, perform some random rotations, determine which of several buckets your normalized vector ends up in, and treat that bucket as a bucket for sorting. So there's different varieties of locality-sensitive hashing. This one's based on angular rotations. There are others based on simply identifying whether or not a given input vector has a positive or negative correlation with another vector. So it's essentially random projections that allow us to that have this property We can go from a high dimensional space to a low dimensional space and still have similar vectors in the high dimensional space appear close together in the low dimensional space, or frequently occur in the same bucket. The concept from the reformer that I think is actually most important. So personally, I don't think that the locality sensitive hashing approach will necessarily stick over time, but this is a beautiful idea inspired by residual nets that really allows us to reduce our memory use during training. And I think this is extremely valuable for training longer sequences. So if you recall residual networks, we represent the output of a layer as the sum, or the normalized sum, of your input plus some function of x. There's a related concept called reversible neural networks, where you have two separate equations. First one being y1 is equal to x1 plus some function of x2. Second being y2 is equal to x2 plus some function of x1. And it has this special property that we can reconstruct fully the gradients with respect to our inputs from the gradients with respect to our outputs and some amount of recomputation. So I won't dive into the math, but just take for granted that we don't need to store more than a single layer's worth of activations at a time. And this is absolutely awesome because it saves us a factor of model depth in memory use during training. And importantly, it doesn't seem to impact accuracy. So this requires writing a custom backwards pass operation for your layers. For the reformer, that structure looks like this. And these are just kind of arbitrary chunkings of your input vector, x1 and x2. So here, x2, we apply the LSH attention block, and that is equivalent to f1. We then sum that with x1. This is kind of like our residual. G in the prior slide is the same as our feed forward block. And then we apply that with x2. We have another residual here to output y2 and y1. So the special structure is just allowing us to save memory use during training. Key ideas here being the concept of reversible networks, the fact that tying keys and queries doesn't seem to negatively impact model performance all that much when you measure in terms of perplexity, and this idea of LSH bucketing. And the reason this LSH bucketing works is simply that the, like I said before, we are preserving the property that things that end up in similar buckets have high attention activations. And finally, we have the routing transformers. This is a paper by Roy et al. And it's quite a similar idea to the reformer, except instead of locality sensitive hashing, they choose to apply a k-means update rule. They also take some inspiration from a paper we discussed earlier, which is the sparse attention paper from OpenAI, where they use this idea of local attention. You can think about it as being attention convolved across your input sequence. So they find that this is a pretty good inductive bias for language modeling to capture local information. And then based solely on the content of each token's, based solely on each token's content, they also apply this routing attention operation. Here's just a little GIF of k-means updates. And one thing you'll notice is that the size of each k-means bucket isn't necessarily the same. For the purposes of the paper, they just, instead of applying k-means in a proper way, they select the k closest values, or the, let's use a different variable, we'll say the m closest values for each cluster center. And this is just to make sure that we have matrices that we can easily parallelize. So if we had matrices of varying widths, we couldn't easily batch our operations. So just like normal attention, we project our hidden state to queries and keys. We randomly project our queries and keys with a fixed orthonormal matrix. Alongside our gradient descent operation, we run a k-means clustering on the projected vectors. So this is separate from SGD. We're applying the k-means update rule on these projected vectors to get new cluster centers. We attend only within each k-means cluster. So in practice, we're attending only to a fixed number of closest terms to each cluster center. And this allows us to, instead of having a O(n) squared operation like dense attention, use an attention operation that is now O(n) by n over k. And if you select optimal k, this becomes O(n) square n, just like sparse attention. So the intuition here is that you have a large dot product between two tokens. That probably means that those tokens are likely going to belong to the same cluster. And if your attention weights are dominated by a few key elements, this means that attending locally within these k-means clusters is a pretty good approximation of what dense attention is doing anyhow. And typically, we do find that many of the attention matrices empirically in BERT are dominated by a few large terms when we inspect the attention weights. So the key ideas here are, again, this application of local attention in combination with a global attention operation based on k-means clustering of the content of each token. So in terms of takeaways, what I hope you got out of this talk, memory use matters, and practical engineering tricks to reduce memory use can have pretty big impacts in the kinds of models that we can train. Checkpointing gradients is kind of an underrated trick that you should probably be applying to your own models if you're not, to get larger batch sizes. If you have expensive operations, see if you can factorize them into the composition of two smaller operations. Another alternative is seeing whether you can compress large matrices into smaller matrices, or cluster information together to use a kind of structured sparsity that approximates the full dense attention. And finally, inductive bias matters. Flexible inductive biases aren't always beneficial, and oftentimes taking advantage of our own human intuition for what is required to solve a given task can allow us to really make some big efficiency gains. And although I didn't really have time to talk about these during the course of this talk, I would also really recommend taking a look at sparse sinkhorn attention by Yi Tae. So sparse sinkhorn attention uses the idea of differentiable sorting to approximate dense attention. So if you're familiar with the idea of sinkhorn iterations or Earth move resistance, they use those concepts for sparsity. There's also a paper called Large Memory Layers of Product Keys that uses another form of factorization to query large sequence independent memory matrices to recall the equivalent of world knowledge or facts that they can use in sequence modeling. And finally, there is a paper called Adaptive Span Transformers that dynamically varies context size per head to see speedups in terms of how much computation is required, particularly on CPU, because it's harder to parallelize their method and apply it to GPU. That's all I've got for you today, but hopefully it was interesting. Hopefully it provided some exposure to some concepts that you hadn't been exposed to before, and gives you at the very least some key terms to search if you're interested.
That was amazing. I feel like that was such a calm and detailed explanation of these papers that I need you to explain all of the machine learning papers to me, and I'm sure people feel the same way. All right, so let's jump into some questions. I see a lot of people saying how much they love the talk and how much they love the slides and the GIFs. I don't see questions yet. Oh, I see one question. Someone was like, I'm surprised you left out the long former. Was there a reason behind this?
The initial reason was just that that particular paper was introduced after I wrote the blog post that this talk was based on. but I do think it's a promising approach. The thing I really liked about the Longformer paper is that they modified an existing pre-trained model to accept longer context lengths. So if you're an industry researcher like me at a small startup where we don't have infinite compute budgets, they lay out a practical approach to make modifications to existing models to solve a specific task, in this case, using longer term context. I thought that was super cool, and that's pretty atypical for papers in this space.
Are you going to do a follow-up covering new things like every few months, maybe?
I'm hoping to update the blog post that I wrote a while back on pragmatic.ml to incorporate some of the newer concepts as those papers come out. So
Nice.
ultimately, my goal is just to find papers that I can apply in my day job, working on document understanding. So,
which actually ties nicely into my question, which is, would you like to tell people about your startup? Because I think your startup is pretty cool.
Yeah. So Indico Data Solutions is a company that builds software tools for non-technical users to use. In particular, we want to give subject matter experts the ability to interact with the machine learning model to automate painful and repetitive portions of their everyday tasks. So everything from processing invoices to pulling structured information out of loans, to one particularly interesting scenario is that we help folks judge whether or not dumpsters are overfull in their computer vision workflow.
Interesting.
Yeah.
Who's paying for that? The government, I guess.
A garbage company.
That makes sense.
Yeah.
Interesting. I see another question. Leonardo asks, do you have empirical results from the techniques that you covered?
Yes, I wanted to include them. It's a bit hard because some of the data sets that folks benchmarked on didn't overlap or are kind of unfortunately small to get reliable results. I'd say the papers that had the most impressive empirical results were the compressive transformer, the sparse transformer, and the routing transformer. I think the reformer paper was a little bit of an earlier work, and I expect follow-up to do a little bit better, but yeah.
Thanks. Charles?
I had a question. Yeah. So this is a little bit broader. I think, yeah, this is super cool stuff. And like making models faster is like something, yeah, I'm not very good at, and I'm glad there are people who are. But I wanted to ask, I guess, like a broader question. Like a lot of this attention stuff seemed to be about trying to make sure that the attention could hold like as much of the document as possible in memory a lot of the time, or like computational tricks to be able to recreate that information later. But when I read something with tens of thousands of tokens in it, right, like a very long essay or a short book, if I need to remember something from the beginning, I notice that I don't have the information I need, then I search back through the text. And that's something that I think a recurrent network would be better at. But I'm just curious if people have thought about sort of, I don't know, maybe like multiple passes of attention or a sort of more active attention mechanism, more akin to like an eye scanning something like is used in computer vision, and less akin to these attention mechanisms that seem to be like, universal.
Yeah, I'm blanking on the name of the paper, but there was a recent paper that was actually for a knowledge plate, a question answering task that first attended over representations for full documents and stored kind of like cast representations for each of those documents, and then specifically queried for relevant documents and looked within those documents to gather facts and information that might be used to answer a given question. So I'll find the name of that paper at some other point, but people are certainly messing around with ideas of attention after the fact, I suppose, is a good.
Yeah, it also sounded, some of the stuff that you presented in the middle, that where you had like routing attention and things like that, those were somewhat akin to like a multi-scale attention where you know that you have to attend to something in the past. So that's definitely similar. But it seems like something that's a little bit more in the like RL, RNN world than the sort of more CNN transformer world.
And some of these approaches even use something that's a little bit more heuristic-esque, just like a nearest neighbor search to find relevant information to use later. Yeah, I think one thing I didn't mention was this idea of hierarchy. Hierarchy is another way to approach problems like this. So for instance, BERT LSTM was mentioned earlier. That would be an approach where we fix higher level input features and have a secondary layer of modeling on top of fixed features.
All right. Thank you, Madison. That was amazing. And thank you for staying up. I'm so sorry I did not realize that.
Thanks for having me.
So bad. Of course. All right. I always imagine people are giving the speakers a virtual round of applause after every talk. So, you know, we're going to insert that here in the recording. But next up, we have Stacy. Stacy is amazing. She's one of my favorite people at work. Don't tell my team I said that, but I love how much she cares about her work. She's a deep learning engineer and she's super smart. Previously, Stacy worked at Flickr, where she worked on a lot of really interesting computer vision applications. And today, Stacy is going to talk about benchmarks, which are, as you can see, actually, I'm going to let her introduce what benchmarks are, but she's going to talk about, in short, collaboration around deep learning using benchmarks. So Stacy, welcome.
Hi, great. Thanks for the intro. Can you see the slides?
Yeah.
Awesome. Yeah, so as Lavanya said, I'm Stacy. I'm a deep learning engineer at Weights and Biases, and I'm going to talk about benchmarks today and how they can hopefully support collaborative deep learning for our planet. Just as a quick outline, I want to focus on Drought Watch because that's the most recent benchmark. That's the one I've been thinking about the most. And from there, expand into new and upcoming benchmarks, talk a bit about the collaborative features that benchmarks enable, and then how you might participate, and then get into Q&A. So I can also adjust depending on what kinds of questions folks have. So jumping right into Drought Watch, and this is work with Andrew Hobbs of UC Davis. And it actually started as his PhD project in which a data set of over 100,000 images, satellite images of northern Kenya was annotated by experts who live in that area to indicate the level of drought at the location. And the reason this is a useful and important data set is that drought is a serious and increasing problem in northern Kenya in particular, but in lots of regions around the world that's worsening with climate change. And there's an existing program in Kenya started by the International Livestock Research Institute in 2011 that uses index insurance, or a measure of, in this case, how bad the environmental conditions are, to pay folks, farmers, or in this case, nomadic herders, relative to how bad the conditions are that year, so that they can use the money to support themselves and their families and compensate for the livestock loss, buy food, etc. And this program has been running for, I guess, 9 years now, and it's expanding to other countries. And it's working pretty well, except when there's a difference in the prediction and the actual drought, then that can convert to a large cost in human terms, based on, either farmers not getting as much money as they should, or the wrong individuals getting the wrong payout. So yeah, as I said, this index-based livestock insurance currently monitors drought conditions using satellite. When drought occurs in Kenya, the program transfers resources to pastoralists, which is the name for these nomadic herders with mobile money, and then they can use that to cover household expenses or livestock needs. And I have here all the wonderful folks who supported the data collection for this, which is Cornell University, Atkinson Center for Sustainable Future, and ILRI. The the issue with the current method is that it's pretty good, but it doesn't actually differentiate between edible plants and non-edible plants from satellite. The current method uses normalized difference vegetation index, or NDVI, which basically compares the green, how green it looks from a satellite image. But as you can see, the problem is that this doesn't distinguish between edible and inedible plants. And the idea and what led to the creation of this data set was to crowdsource expert labels of forage quality and then use computer vision to assess the drought conditions better. So what does the data actually look like? We have ground level labels, and these were actual folks in Kenya who walked around to the location with an iPad and labeled what the conditions look like on the ground at a particular geolocation with a scale from 0 to 3 for how many cows that location can feed. 0 being none, total drought, and 3 being 3 or more cows. So that's really pretty good forage quality. And they also took some ground level images. So we have those for validation of the overall data set. And here you can see some examples of what that might look like in practice and why it could be hard to tell, just by looking at the amount of green that's in the satellite image. So that's the ground level labels. Those are linked with Landsat satellite images from the same place and time. And there's higher resolution and more precise satellite imagery available now, but not at the time that this data set was collected. So potentially with improved imagery, we could improve the accuracy of the model. And the goal is to use only these satellite images to output the prediction or the drought score, 0 being drought and 3 being really good quality. And you can see from some of these example images that it's pretty hard, and the model is actually only looking at the center region in these examples. So that's an overview of the data set. Now I want to get into how we frame this as a benchmark. So initially we launched it in August of last year, right around the CVPR conference. And since then, so I guess it's been about 9 months. Aside from the original authors, we've had 9 participants log over 2,500 experiment runs to the benchmark. The validation accuracy, which is the metric that we're using to compare models, how much better can we do using deep learning methods on this data set? And it's a fixed data set, so comparison is easier. That's been the improvement just from community submissions. And one particularly cool outcome is that there's a paper going into the Vision for Agriculture workshop at CVPR 2020 on climate adaptation or reliably predicting from imbalanced satellite data by Rachit Rawal and Prabhu Pradhan that cite the Drought Watch benchmark and the data set. And this is an inspiring indication that folks are actually building on the existing benchmark data set, looking at the code and using the models to support and accelerate their research, which is really what we want to encourage with these projects. And of course, we're just getting started. I'm going to jump into a report shortly, and here's an overview of what we've done to improve the performance. Basically, at the very beginning of this project, I trained a very simple convolutional neural net to predict, given the satellite image, whether it's from 0 to 3 on the drought scale. And an ongoing task is improving the performance of this model, which includes exploring different architectures, tuning different hyperparameters, also looking at data augmentation, because in the original data set, for example, there's 11 spectral bands for every satellite image, and some of those are noisy because of clouds or other factors. So looking at the variance in that data, perhaps eliminating some spectral bands, filtering out the clouds. There's a class imbalance, so about 60% of the data set is actually drought, which makes it harder to train a balanced model. We also have those labeled ground photos that I mentioned, which we could potentially use to label more data and cross-reference them with the satellite imagery. And finally, like I said, we only have a label for the very center of the satellite image. And the satellite image itself actually covers a much larger area than we are able to label. So perhaps by clustering those regions of the same size, we could label more of the off-center pixels in the satellite imagery. And I'm going to try to jump into the report that I made. Does that still show up?
Yeah,
okay, great. Yeah, so this is a report covering, sort of summarizing the work that's happened on this benchmark. Again, an overview of the data, and you can jump in here with really clear code on how to get started. I plotted the validation accuracy for different submissions. So you can sort of see that the improving trend over time here. There's a lot of different submissions. I went through, and you can actually click through the different submissions that folks have made. The current leader is this user Ubamba98 with a 77.8% validation accuracy, and that was using EfficientNet. And you can, from the benchmark, click into some of these approaches and see, how folks are trying to improve on the baseline model. So it looks like using extra convolutional and dropout layers helped. Fine-tuning ResNet 50 helped in this case. And also Rick137 tried Inception ResNet v2 with custom activation functions and a clocked learning rate. And we can sort of plot this improvement here over time based on when folks submitted. And 2% is definitely an improvement, and these are folks who just decided that this data is cool and they want to work on it and either use it to publish their own papers or just contribute to improving this concrete model. And there's a lot of room to keep making it better, because even these very sophisticated architectures are still pretty close to the original baseline. Here I kind of cover how I developed the original baseline, which is just a nice use case of reports to say, here's what I tried in this phase, here's how I finally got to an improvement. For me, adding class weights because it's imbalanced, and then actually removing some of the channels that were noisy increased the accuracy by 15%, which is pretty amazing. And then I have also set it up with a hyperparameter sweep, which is an awesome feature you can try to just explore what some of the combinations might be that improve validation accuracy. And then you can, see which regions of the hyperparameter space correspond to better performance. So here, it looks like learning rate pretty much varies. Maybe L3 size we want to keep a little lower, and so on. You can also check, what the worst performance is by selecting this one run. And there's lots of ideas for what to try next here at the end of the report. And at this point, I'll hop back to the slides. Oh, I guess one other thing I wanted to show is that you can log the actual examples in weights and biases, and you can see the model's predictions here on the training data. Or sorry, the validation data, but you could log either one. Yeah, so hopping back, an aspect that I haven't touched on yet, those were sort of the concrete details of what this problem is like and what we're trying to solve. Right now, Andrew is using historical data to estimate the economic advantage of deploying a model with this improved index. So how much better would the predictions have to be, how much more accurate the model would have to be, and how that would translate to improvement in terms of payouts to the farmers, etc. And the next step is partnering with ILRI and Takaful to actually deploy this model. So ideally to replace the current model that uses the normalized difference vegetation index and really close the loop on letting the broader world have access to this improved accuracy. And that's a really important component of benchmarks, is that we want to see the solution actually deployed in the real world. In terms of the research contributions, there's a lot of potential in this project to aggregate with other data, for example, other crops like maize, cassava, rice, use it in other regions, whether that's nearby Kenya or potentially in completely different ecosystems. Fine-tuning across several models and transfer learning might do better than starting from scratch. And then this generalizes to any agricultural metric. Basically, if you have any ground level measure or index that's geo-referenced, like a certain crop yield at a particular location, how much visual space the crop takes up, maybe how much of it is affected by a certain pest, etc., then you can use a similar model to basically make a prediction of that metric from satellite images alone, which means you don't need to send folks out into the field. You don't need to do a much more expensive labeling process, provided you can train this initial model. I will take a moment here to jump into the other benchmarks. I know we've been focused on Drought Watch for concreteness, but there's a lot of them. You can find them at wandb.com/benchmarks. The other really big one that we did in partnership with GitHub is CodeSearchNet, which looks at programming code language in 6 different languages, I believe, and tries to learn a mapping between that and comments from a bunch of open source code that GitHub has shared. And that lets you do things like search in natural language for a query like, how do I start a web server, or how do I copy a file, specify the language, and then go directly to the code that does it. And that's just one of the applications. The ultimate goal is understanding code the way that we understand language. And there's been a lot of activity on this benchmark. This is just the overview. You can see in the leaderboard, if it updates, that there's tons and tons of submissions to this one, and we see them coming in. So that's a very exciting other benchmark you might be interested in. There's a space for discussions. There's also a ton of other benchmarks. There's another one with aerial data, which is this aerial segmentation by drone deploy, where we have images from drones, so drone footage shared by drone deploy, where we're trying to label what the drone is seeing. So in this case, you might get an image like this along with an elevation map. And what you want to label is, these are houses, this is vegetation, maybe this is a body of water, etc. And again, this comes with, a really nice data set, nice code to read it in, and instructions on how to get started and train the baseline model. But of course, you're not required to use any of that. So folks have, written completely their own training script. You don't have to use any of the FastAI or Keras frameworks. And it's exciting to see how folks have gone on to improve on the initial baseline that we supply. Yeah, and on that page, you'll see a bunch of them. We try to go for a variety, but as I said, we're just getting started. So a lot of them are vision-based. There's a really cool one where there's a spoiler alert for the Witness game, but that can be a fun way to explore vision without giving away the spoiler. And we're hoping to keep adding a bunch of these. One that I'm working on right now is parsing structured data for campaign finance receipts. So that entails from plain text figuring out, parsing it as a receipt, meaning who paid how much for what ad. And currently that data is required to be publicly released, but it's not required to be easily readable. So there's thousands of these receipts that are readable by a human, but not in aggregate by a machine. So that's an exciting benchmark that we hope to have up soon. And in general, we hope that these benchmarks are useful for learning new skills as they walk you through a project, give you a good data set to work on, maybe a good excuse to try a new framework. see what other people have tried. It's good for reproducibility when it's written up this way. And I know I haven't shown the exact code, but it's easily accessible from any of these benchmark pages. The idea is that you can just step through the instructions and immediately start training a model. And it's already set up to log all of the relevant hyperparameters, config, metrics to weights and biases. So you can both see the life cycle of someone else training their model and their workflow. And you can structure your further work on top of that instead of starting from zero. I'm excited about the real world impact of these benchmarks, and I'm really trying to find more that are focused on environmental sustainability, climate change, positive social impact, but really the application is useful for anything. I think any distributed team with shared goals can structure a benchmark, say this is the metric that we care about, here's all of our different models that we're training, how we're approaching it, and using the existing weights and biases features to basically collaborate on that. And some ways that you can participate, if you'd like, is you can join one of our existing benchmarks. You can definitely start a new benchmark if you have a data set that you're excited about. You can email me with any benchmarks that you think would be cool. And then I want to generally encourage everyone as they do this or as they work on any machine learning project to share intermediate results. I notice that the more folks write up their approach and describe what they tried and what they're doing, the easier it is for everyone else hopping onto the project to catch up and know what's already happened and not be reinventing the wheel. And I think collectively, the more we do that, the faster we'll coalesce around solutions to these important problems. Yeah, and then just growing our community. I'm sure a lot of you are already in the Slack, but here's the link again. I think finding collaborators there, finding projects to work on there, getting ideas is one way that we'll continue to build up solutions to these. Yeah, and that's all I have. I'm happy to take questions, jump into any of the details, and yeah, again, if you're excited about benchmarks in any form, please email me. We're just getting started.
Thank you, Stacy. This is what I love about Stacy. The rest of the company is like building products, writing code, and she is like actually building things to do good in the world, which, you know, is very nice. And we appreciate that you put so much thought into every single product that you build and you think about its impact into the world. And that ties into the first question from Michael, which is there a government or nonprofit entity that your team is in contact with to put the findings from Drought Watch to good use to save lives?
Yeah, so that's all through my collaborator, Andrew Hobbs, and the International Livestock Research Institute, and Takaful is, I think, the other company. He's also happy to answer questions and excited for more collaborators. As he's finishing his PhD thesis, he's going to transition into full-time making this work.
Nice. And I have a question. So what problems make for good benchmarks? So, you know, if people are thinking about pitching you a benchmark, what should they?
Yeah, that's a great question. I think what we really need right now is to see more examples of collaboration. So anything where you think existing approaches haven't worked that well, or you really want to invite more perspectives. I mean, really, any benchmark is good, but I think the more we can see how this tool actually enables folks to collaborate with each other, as opposed to maybe just, you know, one person solving the whole thing right away, that would kind of be less interesting because we don't get to see the evolution of ideas in the space of possibilities.
That makes sense. And I know we've talked about this internally, but people might have questions. How is this different from Kaggle? Because you think about this so differently, right?
Yeah, so thank you. One thing that gets me about Kaggle is a framing where, you know, all of the code is secret and there's a certain deadline until which everyone works competitively to develop their solutions. And then there might be a collaborative phase. And of course, there are, you know, non-competitive framings of this. But what I'm really excited about with benchmarks is making it inherently collaborative and hoping that folks are motivated by solving the problem and by learning more about the space and working with each other than they are by, you know, the prize money or just the sort of the recognition. Although I'm hoping to get recognition in here too, ideally even in a way where it's not just like, these are the lines of code I got and this is the improvement I made, but here's how I managed to explain my ideas. Here's why, you know, I thought this approach might be helpful, because I think that part is really missing from just submitting the code that works, or just, you know, having some model that does something on the validation set that might be really good, but then we don't know why it works or how to transfer it to other contexts.
That makes sense. All right, does anyone have any questions for Stacy before we wrap this up? Someone said they joined late and missed almost everything. That's okay, because we will be posting. Thank you for still joining us. We'll be posting all the recordings on our YouTube channel. And Kayla, maybe you can pop a link to the playlist in the chat. All right. So thank you so much, you guys, for coming. Sometimes we go for 3 hours, sometimes we go for 2 hours. It's always like a good time though. And we've been here for 2.5 hours, and I appreciate your patience. I appreciate Madison, who's been up for like, I don't know what time it is for you, but it seems pretty late. Thank you, Costo. Thank you, Charles, for your thoughtful questions as always. Thank you, Stacy, for being a better person than I will ever be. And still being a badass machine learning engineer. And Tyler left, but his talk was amazing too. And thank you, Kayla. Kayla is so fast, like she will take the stream and split them into recordings that we then share with like over 20,000 people. So thank you, Kayla. This would not be possible without you. The next step that's going to happen is, in case you miss one or a couple talks, we are going to continue to promote them over the next few weeks, so you can catch them there. The speakers will be, we'll link the Twitter information below the videos, but also they'll be in the Slack, so if you have more questions, ask them. Thank you so much for joining us, and we'll see you not the next Tuesday, but the Tuesday after that.